Welcome. Today I will present to you a universal workflow to monitor animal and plant biodiversity using environmental DNA metabarcoding. Biodiversity loss is obviously one of the big problems of our time. However, to detect loss of biodiversity, we need good tools to monitor it. Environmental DNA can be a powerful method to complement classical biodiversity surveys. First, I want to talk to you about eDNA metabarcoding, what it is and how it can be used to monitor biodiversity. So on the left here, you see a typical workflow of an experiment that you would do. First, you would collect samples, environmental samples that could be water, soil, feces, or insects, for example. The second step would be DNA extraction, and then a specific region of the genome called a DNA barcode will be amplified, and this region is informative for phylogenetic analysis to identify the species that this DNA was derived from. Next, this is sequenced, and the sequencing reads are sorted into the different samples again, and this enables taxonomic identification of the different species contained in each of the different samples. This is a method that is being used already worldwide, as you can see here in this overview generated by Compson et al. in 2020. And you can see that biodiversity monitoring using eDNA is already a reality. However, there's still some challenges that it faces to become the global tool to measure biodiversity. And we started by coming up with kind of a wish list of things that we would like to improve to make it more useful for the community. One of the things is that we would like it to be a universal protocol, meaning it can be used for broad detection of species from plants to animals in the same assay. And the same protocol should be able to deal with different types of input. Secondly, we wanted to develop a portable solution that can be deployed in the biodiversity hubs in the rainforest, for example, by local researchers. We wanted the protocol to be relatively fast and simple for the reason that this would also make it more likely that it can be used not in a research institute, but without the need of specialized equipment in the field. And then finally, a big focus of ours was also to increase the throughput of current protocols to be able to process many samples at a time, which is something that would be necessary to monitor biodiversity in a big area like a tropical rainforest, for example. So we started our development from two existing protocols, one called TaxSteady, which is a wet lab method to do eDNA metabarcoding, and a second one, a computational pipeline that uses real-time analysis of nanopore sequencing reads and identifies species more or less in real time from metabarcoding libraries. So from these two protocols, we started to build our own method that is independent of the input type as much as possible. It is highly multiplexed. We combine barcodes for plant barcoding and animal barcoding in the same reaction, making it possible to detect both of them from a single reaction. And then finally, we implemented some workflow improvements to make the whole protocol more high throughput and easier to do. And of course, we changed the typical version of TaxSteady, which is Illumina-based, to a nanopore-based sequencing. As I said, one of the goals was to make it a universal protocol. So the first step that we did was identify primers that we could use to detect both animals and plants at the same time. And while for plants, we found that ITS2, our ribosomal DNA-derived barcode, works pretty well. For animals, we decided to use cytochrome oxidase 1, or also known as COI. And it is known that a big problem with that is that it amplifies a lot of bacterial reads. And so in the end, the information that you guys get is limited. So we tried to optimize these primers to increase specificity and still remain with the same sensitivity. And we did this by combining different already published primers and achieved, as you can see here, a relatively high on-target rate of over 40%, while remaining rather sensitive to detecting many different species. And we did this with the two primer pairs shown here. Next, we wanted to make sure that our protocol works with nanopore sequencing. And so we adapted the protocol and sequenced 4 soil samples, both with Illumina and nanopore, and compared the detection of species. And this is what you can see over here on the left, both for COI and for ITS2. We see that with nanopore, we detect a similar number of taxonomic units on the different levels, species, genera, and families, and can confirm that this protocol works similarly with nanopore. So in the end, we had a very portable solution, and here you can see me and my colleague Esther Lizano in a cabin in the rainforest performing this protocol with a minimal set of equipment that you can see listed here. One crucial improvement that we did to a typical workflow was the way we normalize the libraries. Typically, library normalization is achieved by measuring the concentrations and then adjusting the input for each sample based on these concentrations. This is pretty cumbersome and needs specialized equipment. So we opted for beat-based normalization, meaning we just do a beat cleanup with typical spry beats, diluted 1 to 20, and this limiting amount of beats results in a normalization of the libraries. And here you can see a test experiment where we started with inputs of 500 nanograms to 5 nanograms, and after the beat normalization that you can see on the left, we ended up with relatively similar amounts. We then proceeded to sequence this, and also here saw that the number of sequencing reads that we got per sample were relatively comparable, and the variance that we saw between different samples was comparable to the one that we get when we do concentration-based library normalization. So finally, the protocol that we came up with manages to process 30 samples, including 3 PCR replicates each, in under 9 hours. And this is in field-based conditions, so in a well-equipped laboratory, even more would be easily feasible. And then the second part of this project was to also develop a computational pipeline that can handle the inputs that we get from the protocol that I just presented. And here we, as I said earlier, started from this existing pipeline called NanoRT-AX and developed our own pipeline that we now call RT-Bioscan. And the main features are that it is a real-time analysis pipeline, so the sequences that come from the nanopore sequences are directly analyzed. It can handle multiplexed input data as we would get from our method. And we even were able to use the highest accuracy base calling that nanopore provides for these for this real-time analysis. And in the end, build a consensus sequence from all of the high-quality sequences that we get. The way we managed to achieve this is by using the different kind of base calling algorithms on different levels of the taxonomy. First, we used fast base calling on all of the reads. Based on that, we selected only the reads that were on target, so that were not bacterial, etc. All of these reads we base called again with the high accuracy base calling, which is a bit slower but has higher accuracy as the name suggests. And then based on the high accuracy base calling, we built clustered the reads into operational taxonomic units. And from these, we selected the most relevant sequences and called them with a super accuracy base calling. So in the end, we end up with highly accurate consensus sequences while still being able to process the reads in real time. And here you can see this over time. So you see the sequencing output, you see the fast base calling that keeps up with it pretty well. And then the high accuracy and the super accuracy base calling, which is fewer reads, but just the relevant reads in the end. Here you can see the effect of this. So from left to right, you have the different base calling levels, and you can see how the quality increases with each of the different steps, and also how the length becomes more consistent. This allows us to then really track species identifications in real time while sequencing. And for example, in this case, you can see after 5 hours, we're almost reaching saturations, and we don't detect many more taxons, and would probably stop the sequencing run at this point to save reagents and be most efficient. So overall, the main innovations of this workflow are that it's from sample to identification, one workflow. It's very portable and ready to be used in the rainforest, for example. It is very scalable and can handle up to 96 reactions in one experiment that can be processed in only 6 hours. And one feature that I didn't mention is that it can also do barcoding with the same protocol, which then helps to also generate barcodes from species that are not yet in the databases. And as for future developments, we would like to add additional barcodes to better identify vertebrates like 12S and 16S. We would like to add a feature where we can integrate the barcodes that we sequence into the database in real time, and then use that directly for identifying these new species in our metabarcoding samples. And then finally, we would like to leverage Nanopore's long read capabilities to improve the sensitivity and potentially even go below the species level and detect differences in different populations. And we already started working on that. And currently we have a long-range PCR set up with this 4 kb fragment, including ITS1 and ITS2. And with this, I am at the end of my presentation. I would like to acknowledge all the colleagues that worked with me on this protocol and all of the working group. And with that, I thank you for your attention, and I'm open for questions.